All right. Now, what is the connection between genetics and immunity? What is that connection? Now, genetics is going to be about genetic makeup. You understand that. Very good. Now, you understand that. So, when we talk about makeup, genetic makeup, though, we are talking about DNA, right? That DNA has to be converted ultimately into what? Into proteins. And the proteins are the one that are going to make up a major histocompatibility complex. The proteins are the one that are going to make up antibodies. The proteins will make up the complements that we find in the plasma. So needless to say that all the proteins that are involved in immunity boil down to what genes one has, right? So there is that connection. And then we are going to dive a little bit into the details, right? So proteins are required to carry out an immune response, right? An immune response is going to allow our body to attack pathogens and cancer cells. That, it is something that you already understand from the discussion we've had so far. So, transplanted cells are also going to be attacked, right? But if we transplant the cells, there is a reason, right? So we will have to find ways to interfere with our immune system so that, the transplanted cells are not destroyed, right? So now, as we dive into the different levels of immune systems, we are going to mention that there are two types of immunity. One, it's innate immunity. That innate immunity, it's a built-in immunity, a routine immunity that you don't have to work for to get it. You are born with it, and it can protect you against about anything that you can come across with, right? Obviously, that immunity is not going to be specific, right? So the advantage of innate immunity is that it is immediate and it is generalized, right? It doesn't have any distinction of have you been exposed to that infection before or not. No, it doesn't have that distinction. However, another form of immunity, adaptive immunity, it's an immunity that is very specific. Because it is specific, it takes time to develop, right? And because it takes time, it is slower, right? So the defenses in your body, they are going to act after a line has been breached at the first place, right? So really, your defense is not going to come into play before there is a breach in your skin, in your mucous membrane. And we are going to ask, wait a minute, before we talk about defense, what is our first line of defense? Okay? Our first line of defense is going to be physical, but also chemical, right? So what are these physical barriers that block the pathogens from entering so that, you don't have to trigger your defense system to come on? So, in the first line of defense, we're going to have the characteristic that it is physical and it is chemical sets of barriers, right? These sets of barriers are non-specific. When unbroken, they do their job, right? So unbroken skin, right? We have the mucous membrane and the secretions in the mucous membrane. We have the cilia, that are doing a waving motion, right? The respiratory tract, right? The flushing effect of tears. But not only that, you also are going to have some components in tears that we call lysozymes, right? Lysozymes are able to actually break down the cell wall of bacteria, right? We're going to have tears. Tears are going to also have lysozymes and salt. You have urination, which is a way of flushing foreign particle out. Diarrhea, it's also a way of flushing foreign particle outside. The salt that is in tears and sweat is going to inhibit the growth of microorganisms on our skin. And the oils in our skin is also going to inhibit the growth of microorganisms on our skin. Now, when the first line of defense is broken, then what? Then you are going to need a second line of defense. That second line of defense is going to be of two kinds. You're going to have the cellular component of the second line of defense that involve white blood cells that do not provide a specific immunity. This include neutrophils, monocytes / macrophages, dendritic cells, and other white blood cells, but they do not include lymphocytes, right? Lymphocytes are not part of the second line of defense, okay? However, that second line of defense is going to be part of the innate immunity, right? So now, many cells, including macrophages and dendritic cells, are going to have on their surface receptors. These receptors are called toll-like receptors. These receptors are going to bind foreign particles and internalize these foreign particles that way and break these foreign particles so that they can destroy them, right? So again, innate immune response, it's a generalized defense in the body, and it is produced immediately within minutes. Now, the second aspect of the second line of defense is going to be the inflammatory response or inflammation. Inflammation is when there is a breach. You have a number of components of our tissues that were at one time internalized that are now exposed, right? This is going to trigger a whole cascade of events, right? This cascade of events are going to be very characteristics to inflammation, including, for instance, redness on the skin, the worm in the area where inflammation is happening, the swelling, all of that is going to be caused by what we call vasodilation, or an increase of blood flow that causes a swelling of blood vessels in the area where inflammation is happening, right? So the inflammatory response, it's a central part of the innate immune response when it comes to the second line of defense. So it's going to create an environment that is not very enjoyable for foreign particles. Now, whenever you have a breach in the skin, right, this is going to be an open area for bacteria to get in, viruses to get in, right? And as these viruses and bacteria try getting in or actually get in, right, you need the body to develop a response. The warmth, it's actually going to neutralize microbial growth or chances of microbial growth, right? The swelling that is caused by vasodilation, it's basically that the cells that make up the blood vessels are going to spread apart from each other, which is going to allow white blood cell to squeeze out of the blood vessels to get at the site of potential infection. And when these white blood cells get there, they are going to engulf the foreign particles, right? So, for instance, the monocytes, which are a type of white blood cells, are going to differentiate into macrophages and dendritic cells, and the macrophages and dendritic cells are going to engulf bacteria, viruses, and get rid of them that way. So, we're going to have phagocytic cells, right, that are going to gather at the place of inflammation, and these phagocytic cells are going to cause a destruction of any potential foreign particle, but also restoration of the tissue. All right, so the proteins of the immune system, when it comes to innate immunity, we're going to have the complement. The complements are part of the plasma. This complement are proteins which job is to cause puncture in bacteria so that the bacteria can lose their content and die, dismantlement of viruses, activation of mast cells, okay, that are going to release histamine and attract phagocytes like macrophages, neutrophils. Then we have the selectins. The selectins, their job, these are proteins, their job is going to recognize recognize the surface of bacteria, right? Yeast and viruses and how they differ from self, right? So remember that the system has to recognize self versus non-self, right? And then we have a group of what we call the chemokines, right? Or cytokines. These cytokines are going to be of many different kind. We're going to have the colony stimulating factors or CSF. This CSF, their job is going to be to stimulate the red bone marrow to produce lymphocytes, right? The interferons, they are going to be involved in blocking the multiplication of viruses. They are going to stimulate macrophages so that these macrophages can engulf viruses and bacteria. They're going to stimulate B cells so that B cells can produce antibodies that are going to attack foreign particles and also to attack cancer cells. The interleukins are going to be very important in controlling the differentiation of lymphocytes, right, to multiply and actually get to do their job. And the tumor necrosis factor or TNF, this TNF are very important at stopping tumor growth, releasing growth factors that are going to cause a multiplication of white blood cells and cause a differentiation of white blood cells so that a better immune response can be developed. All right, so we have the innate immunity with the physical and chemical barrier. You have innate immunity with the cellular and molecular barrier here, where we're going to have the phagocytic cells and so on. And you're going to have, if that line fails, the second line of defense, you're going to have a third line of defense. That third line of defense is going to involve the adaptive immunity. In the adaptive immunity here, the antigen presenting cells, so the macrophages, are going to present antigens, okay? And by presenting these antigens, we are going to have T cells that are going to come into play and neutralize the cells that are carrying antigens. So that's one. Two, it's going to be that B cells are going to undergo differentiation and form plasma B cells, and by forming plasma B cells, they're going to produce antibodies that are specifically designed against the specific antigens that are present in our body at that time. After an infection is under control, what will happen is that some cytotoxic T cells are going to be saved as memory T cells, while some B cells are going to be saved as memory B cells for the next time an infection comes, the same infection, actually, a response is going to be mounted in a relatively short period of time. Okay? So, adaptive immunity requires stimulation. So, no stimulation, no adaptive immunity. It takes time to develop adaptive immunity. That is a very important point to remember. And also, what is going to be characteristic of adaptive immunity that I want you to remember? It's that it is very diverse, right? So many different pathogens can be recognized by adaptive immunity, but that requires pre-awareness, right? You need awareness, and that awareness is what builds up the specificity, right? To be able to even tell from one strain of influenza to another strain of influenza, right? We have a memory that is developed as consequence of an infection that was under control, right? Which is going to allow us to develop a faster response. Now, when it comes to memory, we're going to have a primary immune response, which is the first reaction, the first specific reaction to an antigen. And then we have secondary response, which is the second response. That second response is actually the first memory response, right? It's the first memory response. Now, when you have a third boost, right, that third boost is the second memory response, right? Now, the more boost of immune response you have, the stronger that immune response, the better that protection. That's why when we do vaccines, boosts are necessary to provide a good level of protection. All right, so in terms of adaptive immunity, we are going to have two types of adaptive immunity. We're going to have humoral immunity, right? Which is a type of immunity where B cells are going to produce antibody. That's why that immunity is also called not just humoral immune response, but also antibody-mediated immune response, right? So this involves B cells that produce antibodies and T cells, and specifically helper T cells. Now, then the second type of adaptive immunity I'm going to address, it's cell-mediated immunity or cell immune response. That cell immune response involves helper T cells and cytokines and cytotoxic T cells. But in addition to this different components I just mentioned, you are also going to have the antigen presenting cells or APC, right? Okay, so B and T cells are going to start the B and T cells are going to differentiate in the bone marrow. I already mentioned that, right? However, they are going to migrate in different organs, including the lymph nodes, right? And the thymus. However, the T cells are the one that are going to learn in the thymus the difference between self and non-self. All right, so humoral immunity, how does it get developed? So typically you're going to have free-floating antigens. Free-floating antigens, right? These free-floating antigens are going to trigger a response. Response from whom, right? You're going to have that these antigens are going to be recognized by B cells, okay? They're going to be recognized by B cells. These B cells are going to have on their surface B cell receptors. B cell receptors, so B cell receptors, BCR. Now, B cells have different kind of B cell receptors. So here we have one type of B cell receptor with the same type of receptors. Here we have another type of receptor. Here we have another type of receptor, right? And here another type of receptors. We have this diversity of B cells. So the question is, which of the B cell is going to be specifically undergo proliferation for the specific protection against this antigen right here, right? This is when the story becomes important when it comes to humoral immune response. So the B cell receptors with the matching receptors with the antigen are going to be the B cells that are going to undergo proliferation under the control of cytokines, right? And as they undergo proliferation, notice that only one type of B cells with B cell receptors are going to proliferate, right? Some of these B cells are going to become plasma cells. These plasma cells are B cells that are releasing their receptors. At that point, they are not called B cell receptors anymore. They are called antibodies. Okay? So, now, these antibodies are released by secretion, right? And now these antibodies can travel in the plasma and go target the antigens. And as they target the antigens, now these antigens are now in a state that they can be discarded. Okay? Now, after an infection is over, most of the plasma cells are going to undergo apoptosis or programmed cell death, right? As the cells undergo apoptosis or programmed cell death, this is to actually prevent the cell, the body from keep making antibodies for an infection that is not there anymore, right? You have a small fraction, about 5% of all the B cells that are going to stay as memory B cells, all right? So we are going to have a polyclonal humoral response. What we mean by polyclonal humoral response, it's that when you consider an organism such as a bacterium has many types of molecules on its surface. So you have some B cells with B cell receptors that are going to recognize some molecules. Others are going to recognize other molecules and other molecules, so that not only one single B cell with B cell receptors is going to exclusively recognize one bacterium. No. You're going to have multiple B cells with different B cell receptors. Because of that, many groups of B cells with B cell receptors are going to undergo clonal expansion. And because these clones are different, we say that this is a polyclonal response, right? Poly, many, clonal, clones, right? So, which is going to allow us to produce multiple different kind of antibodies, which actually optimizes the chance to control the infection. All right, so now B cells produce antibodies. Now, these antibodies, what do they mean in terms of genetics, proteins, structure, right? So, now we are going to dive into this. Remember, we said that there is a connection between genetics and immunity. But what is that connection, right? So, I'm coming. Now, notice that an antibody has a Y shape, right? It has a Y shape, basically like this, with a foot and arms, right? Now, when you consider the molecule, we actually see that there are 4 proteins. We have 1, 2, 3, 4. Now, these 4 proteins are really classified into 2 groups of proteins. We have the first group right here, which is actually called the heavy chain. Okay? So we have a heavy chain. Chain. All right? And we have the light chain. This is the light chain. Okay? Light chain and heavy chain right here. Now, the 2 heavy chains are going to be connected together through disulfur bonds. Disulfur, this is a sulfur residue and another sulfur residue, right? And this is how the heavy chains are going to come together and the protein is going to be stable that way. Now, the light chains are going to be associated to the heavy chain right here with the disulfur bond as well, okay? Forming this complex structure. Now, with that being said, there is the foot of the antibody is going to have what we call a constant region. That constant region is the same for a given class of antibodies. And there are different classes of antibodies. There are actually 5 different classes of antibodies. We're going to discuss that later, right? We have a constant region. I want you to remember that. And you do have a variable region, right? That variable region, it's what is going to set apart one antibody of a given class from another antibody of the same class. Okay? For instance, antibody IgGs, all right, from another antibody IgG, right? How are they different? Well, they are different because this place where antigens are going to come and bind is going to vary, right? And this is what allows us to actually target different antigens, right? Because different antigens have a different 3-dimensional structure, right? No question about that, okay? So, now, with that being said, the area where the antigen is binding, right, is going to be called the epitope. Actually, no, the epitope is the area of the antigen where the antibody is binding. There we go. Let me repeat that. The epitope is the area of the antigen where the antibody is binding, right? Antigen binding site, right? So this is where the epitope is going to sit, right? This area being the epitope, okay? All right. So antibodies, beside their primary structure, can complex with each other to form more complex structure. We have IgA right here. Now, I was saying antibodies. Now I'm saying IgA, right? What does that mean? Ig stands for immuno immunoglobulin. Immunoglobulin, right? IG. Immunoglobulin class A. Immunoglobulin class M, right? Okay, so immunoglobulin class A has two antibodies attached to each other. Immunoglobulin class M has 5 units, right? So we have 1, 2, 3, 4, and 5 units, right? So different classes of antibodies and their role. We have IgA. IgA is found in the milk, saliva, urine, tears, right? It is found in the respiratory tract and digestive tract, in the secretion, more specifically. Their job is actually to protect against pathogens, right? At the point of entry of infection. Then we have IgDs, immunoglobulin class D, right? These ones are going to be found on B cells in the blood. So they are going to stimulate B cells to make other type of antibodies, and particularly in infants, right? We have IgEs. IgEs are associated with allergic reaction, right? They are going to be found in secretion with IgAs and in mast cells, right? They act as receptors for antigens, right? And they are involved in allergic reaction. How about IgGs? IgGs are the most abundant immunoglobulins that you are going to find circulating in the blood. They are going to protect against bacteria, viruses, toxin, and very specifically against, protecting in secondary immune response, right? For instance, when you have a vaccine, you have a first shot and a second shot. In the second shot, you're going to find a response of IgG that is going to be in huge amount, right? IgMs are found in the plasma. These IgMs are actually the first antibodies to be produced when there is an infection, right? The initial stage of an infection, when you have the first antibodies produced, the IgM are the one that are going to be produced, okay? So the job of antibodies, what is their job? Their job is to attach to antigens, right? And that attachment to antigens, it's to label this antigen so that that antigen can be destroyed, okay? Now, the attachment of antibodies on antigen is called opsonization, right? This allows the system to make more visible the particle to be destroyed, okay? So other thing is that antibodies are going to constitute a response that sometimes can be harmful to our body. For instance, when you have a huge amount of IgE and this IgE actually are just binding to mast cells, this can actually cause what we call anaphylactic shock, right? That can actually cause someone to die because the airways are going to close up, right? Because of that huge inflammation, and the individual is going to be unable to properly breathe, and that can cause the death of the individual. Now, let me tie antibodies and genetics. Now, antibodies are very diverse. And the reason why they are diverse, it's because our body has a way of shuffling the genetic information, not the genetic information itself, but after transcription, what happens with that genetic information? We talked about splicing, right? We talked about alternate splicing, right? So that alternate splicing is going to produce a tremendous variety of antibodies, right? So the genome has a limited number of antibody genes. However, with splicing and alternate splicing, the combinations of antibodies become wide. However, in spite of that, you're still going to have your 5 classes of antibodies, right? Okay? And those 5 classes of antibodies are going to be determined by the genes that are present, right? So in early development of B cells, sections of antibody genes are going to be rearranged along chromosomes, right? And as they are rearranged, this is going to determine, right, the different types of antibodies that are going to be produced. So we have different combinations of the V, which is the variable region, and D, the diversity region, and J, the joining region, so that you have combinations between V, D, and J that is going to cause antibodies to be different from one from another. So here, if you consider the heavy chain, if you consider the genes for the heavy chain, we're going to have V1, V2, V3, D1, D2, D3, J1, G2, J3, J4, and so on. And then we have the C region here. Now, this is on a chromosome, right? Now, then you are going to have gene rearrangement down here. And that gene rearrangement is going to select only a particular V, a particular D, a particular J, right? So that at the end of the day, for the heavy chain, you are going to have this content region with a variable J, D, and V area. What is happening with the heavy chain? The same thing applies to the light chain, because you do have a conserved region right here across the class of antibodies, right? But you also have a variable region right here, right? With the joining and the variable area here, okay? So that you are going to have multiple different types of antibodies. Okay? Now, in terms of cellular mediated immune response, we're going to have to shift our attention to T cells. And the T cells, remember, they mature in the thymus. They learn the difference between self and non-self. We have helper T cells, cytotoxic T cells, right? Okay? So these guys, as they recognize all cells, they are going to provide what we call cell-mediated immunity, right? So what are the cells that are grouped of the T cells? We're going to have the helper T cells that are going to have on their surface CD4 proteins, and these CD4 proteins constitute actually antigens, right? The helper T cells are going to have a bunch of functions, or multiple functions, including regulating the immune response. Then we have the cytotoxic T cells. The cytotoxic T cells are the cells that the whole cell is going to be toxic for other cells. That's why we call it cytotoxic T cells. they're going to have on their surface CD8 markers, and this CD8 are antigens. Now, cytotoxic T cells, because they are toxic for all the cells, killing the cells, they are also called killer T cells. Then we have some T cells that are what we call regulatory T cells, okay, also called Tregs, right? These Tregs are going to make the immune system softer, so they're going to dampen the immune response so that, our body is not too harsh, developing too harsh of a response, because whenever we develop an immune response, our body is actually feeling that and responding to that as well. So the role of helper T cell is going to be essential in humoral immune response as much as it is essential in cell-mediated response, right? In humoral immune response or antibody-mediated immune response, the helper T cell's job is going to be to recognize antigens presented by macrophages and to stimulate B cells to produce antibodies. How about cell-mediated immune response? The job of helper T cells is going to be here to secrete cytokines, which are chemokines, right? And to activate cytotoxic T cells, okay? Finally, the role of cytotoxic T cells. The role of cytotoxic T cell is going to be to attack any cell that is presenting antigens, so antigen presenting cells, right? That are obviously presenting antigens, right? They are not just going to attack a macrophage that doesn't have anything as antigen on its surface. No, they're not going to do that, right? They are also going to be very effective at killing cancer cells. Okay, they are very good at killing cancer cells because these cancer cells are misbehaving, right? So they are going to kill them. But how do they do that? They are going to come in contact with the cell to be killed, right? So the MHC is presenting an antigen. The cytotoxic T cell with its receptor is going to recognize that MHC with the antigen. So they're going to come close together. The cytotoxic T cell is going to actually release some proteins that we call perforins. These perforins are going to poke holes in the cell to be destroyed. And through the perforins, that hole, you have some granzymes, some enzymes that are called granzymes that are going to go from the cytotoxic T cell to the cell to be destroyed. And that way, that cell that is to be destroyed is going to be destroyed by the granzymes. Okay? So this is how they destroy B cells. This is how they destroy macrophages, dendritic cells, cancer cells. Okay? So they are able to kill about any cell that is misbehaving or not behaving properly. So here we have a cancer cell, right? That is recognized with a receptor right here, right? A T cell receptor. And you see some chemicals right here that are the perforin. The perforins are going to come and form holes in here. And what we are not showing here is that, well, this cancer cell is not just going to lay out its content, but this guy is going to actually release some granzymes in this cell and cause a destruction of this granzyme that way. Cause a destruction of this cell that way. All right. So, finally, but not least, let's take a look at the different types of immune system cells. The macrophages, they are going to present antigen. They are going to engulf antigens. The dendritic cells, they are going to engulf antigens, right? And do through phagocytosis. The mast cells, they're going to release histamine in inflammatory response, and they are going to release allergy mediators. The B cells, they are going to mature in antibody producing cells, right? And they are also going to develop memory B cells. The T cells, we're going to have helper T cells, which job is going to be to recognize non-self antigen presented on macrophages. They're going to stimulate B cells, and so that these B cells can produce antibodies. The helper T cells are going to release cytokines, and they are going to activate cytotoxic T cells. The cytotoxic T cells are going to attack cancer cells and cells that are infected, carrying viruses and bacteria. The regulatory cells are going to suppress immune response, while the neutrophils are going to attack bacteria, engulfing them, right? The natural killers are going to attack cancer cells and infected cells so that they can be destroyed. So this is where we are going to stop this first segment of this chapter. Thank you for your attention.